one of the questions that have been raised a couple of times during the last 2 days has been the issue of cell type specificity, diluting out effects that are seen in transcriptional analysis. And so what I've been working on is cell-specific analysis in the Arabidopsis leaves. And so if we look at the Arabidopsis leaf, it's quite a complex organ. There's loads of different cell types in there. And so in order to reduce the complexity of what we had to work with in the first instance, we decided to work only on fully developed leaves, which still contained quite a number of cell types. So what we wanted to do was obtain cell type specific transcriptomes, and the only method that was really available that could be used without any prior transgenic lines and so on is really laser capture microdissection, and in the case of trichomes, of course, you obtain those in a different way. So what we had to do was to get a lot of laser captured samples, which takes a very long time for very little gain, unfortunately. So that's one of the main challenges, was actually getting enough RNA. So as I said, the challenge is in a sample we collected about 500 cells, which means you expect probably around 500 picograms of RNA. And then when you have wax in there that actually have a very negative effect on the RNA you can recover, which means that you're probably talking a lot less than that. So either you spend the most of your life sitting in a basement somewhere in front of a microscope and cut out enough cells, or you go through multiple rounds of amplification. One of the problems with this is that this can affect both the quality of the RNA, but also the relative quantities of the different RNA species in there. So we had to find out how many rounds of amplification we needed to do and how this affected the RNA. So first of all, we found that 3 rounds of amplification is enough will give us enough RNA for microarray if we start from 50 picograms of RNA. And also, if we look at the size of the amplified RNA, we see that it goes down to just over half the size of standard treatment, which is 1 round of amplification from a microgram, but that the distribution tend to be about the same. So the question is, how does this then behave when we put it onto microarrays? So that was the next thing we did. So here it's comparing. So it's comparing the relative expression when it's compared using the standard 1 microgram, or either low or very low, or the 2 low versus each other. And what we see is that about 98% exhibit less than a 1.5-fold difference when amplified 3 times rather than 1 time, and more than 99.9% exhibit less than a 2-fold difference. And that means that if we do this when we analyze our data, we have to keep this in mind, which means that as we go towards 0, of course, we can't really say anything about differential expression. As we move towards 2.25 fold difference, we start getting quite happy, and anything above 4 we are really, really happy with. So for the experiment, we obtained, as I said, all the different cell types, or the major cell types from the leaf using laser capture microdissection, trichomes, using vortexing methods. And then we analyzed these on NimbleGen expression arrays and performed some data analysis using NimbleScan software and the Gaga package for R. And then the question is, if we just put in the data, do we get anything meaningful out? Because that's the first confirmation that it actually is a valid way of doing the experiment. So first off, I took the first 10,000 genes, so that means only genomic nuclear genes. There's nothing mitochondrial or chloroplast in there, and then allowed the program to order this to cluster the cell types based on the transcriptional information from the genes. And this was quite encouraging. We have the mesophyll grouping together here, palisade mesophyll and spongy mesophyll grouping closer together, and then the bundle sheath cells coming in. Also, we have the epidermis, in this case pavement cells, sister cells, and guard cells, with the sister cells and guard cells grouping slightly closer, probably due to a shared developmental lineage. Trichomes are on their own. Also vasculature groups on its own quite far away. Then there's marginal epidermis and hydrothodes, although with those I'm a little less certain about the quality of them because I only have 2 biological replicates for hydrothodes and 1 for marginal epidermis. But it's quite encouraging that they actually group the genes together in the way we expect. So that allows us then to go further on, and then we can look for cell type specific genes, which is what we have here. So in each case you have the different cell types. These are the abbreviations here. So you have the mesophyll up here, you have the vascular, you have the epidermis, trichomes, hydrothodes, and marginal epidermis. And then the numbers are how many genes there are in these groups. So how many, if you want cell type specific ones, there are. And it's not surprising that these almost always, with the exception of in xylem and in trichomes, tend to be up, because what I asked the model to find was genes that are specifically expressed in one way in a cell type, but in exactly the same way in all the other cell types, because that's most likely what would happen if it's off in all the other cell types. Trichomes are slightly different. They also contain the most differentially expressed genes, and that's probably because basically they are almost an organ in their own right, and therefore quite far from the other cell types. We can also look at cell populations, so we can look at the mesophyll together or without bundle sheath. Equally, we can look at epidermis with or without guard cells. And we can look at the vasculature as a whole, in which case it behaves with the number of differentially expressed genes almost like the trichomes, showing that they probably have a very different function from anything going on in the rest of the leaf. So another question then, or the next question that comes is, are these groups, do they give anything meaningful, or is this just random? So if we look closer at one of the groups, in this case it's the mesophyll group, and then do a bingo analysis and look at which GO terms are actually overrepresented in this group, what we find is, again, very, very good. It's what we expect. So it's to do with thylakoids, plastids, photosynthesis, generation of precursors, so precursor metabolites and energy. So to do with photosynthesis, which is what we expect to find in the mesophyll. And also, if we look at cellular components, we again find that it's a lot to do with photosynthesis, confirming that these data are actually are valid. And that allows us then to look at other cell types. So, for instance, in this case, it's trichomes. The first thing we can look at is, because trichomes are very different, they contain lots of genes that are actually down-regulate or down compared to all the other cell types. And one of the things we see is, again, photosynthesis has been shut off, which is not very surprising. And also, it seems like stress response is not something that's happening in trichomes. Again, this is, of course, in the absence of stress, but still, you wouldn't expect to find it there otherwise. And if we see what is up, molecular functions, it's a lot of peptidases, enzyme inhibitors, hydrolases, pectin esterases, and other esterases, which is, again, what you would expect in trichomes, because they are involved, among other things, in physical resistance towards insects and so on. So the question is then, where do we go from here? Because the next thing that we would then be interested in doing is looking at this in a more complex way, so looking at developmental time series or looking at different treatments and so on. And for this, laser capture microscopy or microdissection is definitely not the way you want to go. So there are other methods. depending on what you're interested in. If you want the complete transcriptome, you have fax sorting, which has been used quite a lot in roots and also in shoot apical meristems. Or you can go for the intact system, which will give you the active transcriptome. So this is immunoprecipitation of nuclei. Or you can go for the translatome, which you do through the immunoprecipitation of polysomes. So the various methods have pros and cons. So obviously, a really big plus for the laser capture is that you don't need any markers to do this, whereas for all the others, markers are a necessity, and they just don't exist for all the different cell types. However, if we look at it from the other side, laser capture takes a long time, and it's quite expensive, whereas all the other methods are at least relatively cheap and fast. So what we have decided to do is then work with the fax methods. So based on the laser capture data and other data that we have had from previously, we are generating transgenic lines that then cell type specifically or cell population specifically expresses GFP, and we're using a transactivation system for this. So this is one that was developed in Ian Moore's lab here. It's the LHD4 artificial transcription factor, which activates transcription from the POP6 promoter. In our case, we have GUS and GFP, but because it's transactivational, if we find any good lines, we can actually reuse them later on for other methods. So I can quickly go through this. We have a number of different lines now. We can protoplast them, not very surprising. But what we can also do is we can also sort them even in the presence of large amounts of cells with chloroplasts and autofluorescence in them. So this is important, as I said, when you want to do more complex analysis. So if you want to look at several cell types, you want to look at different developmental stages, you probably also want to look at environmental conditions, and then of course you need all your biological replicates. And again, this becomes way too much to do with something like laser capture, whereas with fax, this is a relatively quick and easy procedure. And the final point I would like to make is that also another advantage of fax over the laser capture is that it allows you to basically take any population that's marked with GFP. So in this case, it's a cell population that's associated with senescence. It may be a bit difficult to see, but I guess that's part of the point, that this is at such an early stage of senescence that you almost can't see it's happening. But if you look at it with the GFP markers, you see that, and you can take these cells and sort them, even though it's a mixed population of cell types, and then you can use these for microarray. So to finish off in a summary, what we have done really is we have managed to create a transcriptional map of the Arabidopsis leaf, which is one of the things that would help with some of the questions that have been on cell type specific responses during the meeting. Also, we are currently extending our collection of GFP lines so we can use these for fax, which can be used, as I said, on any GFP labeled population, which is a very big advantage. Also, we're currently using this to study development and stress. And finally, using the transactivating system, we actually can continue to use these lines. We can even from exactly the same line get whole transcriptome data, actively transcribed data, and what's being translated from exactly the same sample. And also we can do cell type specific or expression and knockdown. And so finally, I'd like to thank the people who have been involved in this. Sanjeev, who has been the technician who has been helping me with all this. Alison, who is doing the work on the fax on cell type specificity. Nihal is doing facts on the senescence. Jim Bain and Vicky Buchanan-Wollaston are the PIs that I've been working for. The laser capture I did in Keith Lindsay's group in Durham, and Jen was a tremendous help with that. And I have been funded by the Agronomics project, which is a project under the Framework Program 6, and is what's it called? Administrated by Pia Hilton at VIB, and Nihal is funded by the Turkish Science and Research Council. So, thank you.